Once we know the different tonicity solutions, we can talk about the differences in how an animal cell is going to respond to those solutions and how a plant cell is going to respond. And the key to how these two cells are going to respond to these solutions really has to do with the exterior of their cell, or what components we find outside the plasma membrane. So just to review really quickly, on an animal cell, on the outside, we're going to have that extracellular matrix. So that extracellular matrix, the key to this is that it is going to be very flexible. So I'll call it flexible here, and we'll also put stretchy because that will define how it responds really in the various solutions. And then for the plant cell, on the outside, we're going to have a cell wall. And in contrast to the extracellular matrix, the cell wall is going to be very rigid and it's not going to stretch really. So let's keep this in mind as we go through the three different tonicity solutions a little bit again. So here we're looking at the hypotonic solution again. The hypotonic solution has a low solute in the solution, and because it has a low solute in it, we're going to have a correspondingly high amount of free water. And again, if you compare that to what you have inside is going to certainly be higher solute. And because it's higher solute, it will be lower free water. And so if we look at the direction that the water is going to actually move, we're going to see that the water moves high to low. So the water will be moving into this cell. That's always going to be the case when we're talking about a hypotonic solution. Now, if we talk about what this means for an animal cell, for an animal cell, recall that the outside of them is very stretchy. So the animal cell will gain water. And when it gains water, it will stretch. Now, how much water moves into the animal cell is really going to depend on how much the concentrations differ outside the cell versus inside the cell, because the water is going to continue to move in there until we reach that equilibrium point. Well, if too much water moves in, these can actually rupture. And when they do happen to rupture, we refer to that process of rupturing as lysing. Or we can say that this is lysis. And if we happen to have a ruptured animal cell, then that lysis or that bursting is going to be a deadly situation for the animal cell. Now, if we talk about the plant cell and contrast it here, the plant cell is also going to gain water. Now, when the plant cell gains water, because the cell wall is so rigid, the cell here is not going to burst. And so it's not going to lyse. And instead, it's going to get really full and it will really just plump up. So it will get plumped. And so if I put that here, we do have an official word that refers to this plumping up of the plant cell. So if we put that on here, the plant cells in this type of environment, they're going to be called turgid. Turgid refers to them being just, again, really filled full of water. And this is actually ideal for a plant cell. They would love to be in a hypotonic solution. So that would be good for them. On the other side, when we're talking about the animal cell, again, if this happens, that is actually deadly. So that's the hypotonic solution. With an isotonic solution, we have the same concentration of solute inside and out. And so since we have the same concentration of solute inside and out, then we expect to have the same concentration of free water inside and out. So what this means for the direction of water movement is that we will have some water moving in, but we'll also have some water moving out. So there's going to be no net movement of water. And so if we compare this between a plant and an animal cell and what the difference is going to be for the two cell types, for the animal cell, the cell is going to stay the same. So no real effects on it. And in this case, this is going to be the ideal environment for an animal cell. So they would love to be in an isotonic solution. That's the good scenario. If we compare this situation in the plant cell, The plant cell is also going to have no net movement of water, so it's going to stay the same size. But in the case of the plant cell, this is going to lead to a really flimsy cell. And so that flimsy cell is really in an okay situation. It's not ideal though. It's not as good as when we had the turgid cell. And in this case, we refer to this cell as flaccid. So a flaccid cell for a plant cell is not a particularly happy cell, not particularly healthy, but it is surviving. It is in an okay situation. This would be similar to like a wilted plant. In the hypertonic solution, outside the cell, we have a high concentration of solute. And if we have a high concentration of solute, that means we have a low concentration of free water. And so if we compare that to what we have inside we're going to have low solute, and low solute would mean high free water. So if we look at the direction of the water movement, water is always going to move high to low, and so it's high here and low here. The water is actually leaving or moving out of the cell. If we look at the effects this is going to have on our two cell types, an animal cell, it is certainly going to be losing water. And so water is leaving, and so this becomes a dehydrated cell. Not a good situation for an animal cell. Now, again, if too much water is actually moving out of the animal cell, this could be a very bad thing. It could be deadly. So it can be deadly. And if we look at the effects of this, the animal cell is going to go from looking like this to actually kind of collapsing in on itself, something like that. This cell is what we would call a shriveled cell. And we can also call it a crenated cell. So the crenated is kind of referring to all of these little folds that we happen to get. That's the animal cell. If we look at the plant cell is also not happy in this environment. It's going to lose water. It's going to get dehydrated. All of that is true as well. But because it does have this different covering on the outside, the plant cell is going to go from something like this, where you actually have the cell wall and the plasma membrane right there next to each other. And then after the water actually leaves, so if water starts moving out, what we're left with is the plasma membrane starts to shrivel up inside the cell. And when this plasma membrane, which I'm going to label it here so we can see, that's my plasma membrane, this is my cell wall. When this happens, they actually separate from each other. And we end up with actual space right there. So this would be airspace. That is a deadly situation for a plant cell. We do have a name for that. We refer to it as plasmolysis, or we say that the plant cell got plasmolyzed. And if you have a plasmolyzed plant cell, again, that separation of the plasma membrane from the cell wall, that cell is going to be dead at that point. So there you have the three different tonicity solutions: hypotonic, isotonic, and hypertonic. And we have the differences between how the animal cell is going to behave in them and how the plant cell is going to behave in them.